last lecture, in the end, I started the discussion on skeletal muscle. I discussed about the sarcoplasmic reticulum or sarcotubular system consisting of the longitudinal tubules or L tubules having dilated ends called terminal cisternae, and these terminal cisternae, these surround T tubules or transverse tubules. So these T tubules, these extend from one side of the muscle fiber to the other. Right? This is a T tubule. T tubule communicate with the extracellular fluid and also contain this fluid. Right? I told you that sarcoplasmic reticulum is concerned with storage, release, and uptake of calcium ions. Storage, release, and uptake of calcium ions. Calcium ions initiate the mechanism of muscle contraction. Calcium ions initiate the mechanism of skeletal muscle contraction. You see, in the sarcoplasm, there are a number of myofibrils. So, number of myofibrils are present in the sarcomere. You see, one of the myofibrils. Right? So these are the myofibrils present in the sarcoplasm, of the muscle fiber. This is the muscle fiber, whole muscle fiber, and these are the myofibrils present in the sarcoplasm of the skeletal muscle fiber. Now, each myofibril is parallel to the muscle fiber. So these are parallel to the muscle fiber. Each myofibril is 1 to 2 micrometer in diameter. So 1 to 2 micrometer is the diameter of the myofibril. And there are a number of myofibril in a skeletal muscle fiber. Each myofibril is composed of 2 types of filaments. So 2 types of filaments constitute the myofibril. Thin filament or actin filament, and the second is thick or myosin filament. So each myofibril is composed of filaments, thin or actin, and second is the thick or myosin filaments. Diameter of actin filament, thin filament, is 50 angstrom units. So 50 angstrom units is the diameter of the thin filament or the actin filament, while 100 angstrom unit is the diameter of the thick filament or myofilament. Right? So diameter of thin filament, actin filament is 50 angstrom unit, and diameter of the thick filament or myosin filament is 100 angstrom unit. Now these filaments, thin and thick, actin or myosin, these partially overlap and interdigitate. To form alternate dark band and light band. So these filaments, I mean these actin and myosin filaments, these partially overlap and interdigitate to form alternate dark and light bands. You see, this is the dark band, it is also called A band, and this is the light band, which is also called I band. I band. And you see, this is the thick filament, myosin filament, and these are the thin filaments or actin filament. You see, red colored, these are the thin or actin filament, and these are the myosin or thick filament, and these are partially overlapping, and these interdigitate to form alternate dark band, also called A band, and light band, also called I band. You know, a skeletal muscle has transverse striations. Transverse striations or cross striations. Transverse striations or cross striations. Now these transverse striations are due to these alternate dark and light bands. These are due to alternate dark and light band. Dark band, I told you, also called A band. A is from anisotropic. Anisotropic. It means having unequal refractive index to polarized light. So unequal refractive index to polarized light. And what is the polarized light? You know, light is projected in all directions. And when it is projected in one direction, it is called polarized light. So A band is from anisotropic, which means unequal refractive index to polarized light. Light bands are also called I band. Because these are isotropic to polarized light. Isotropic, having uniform or equal index, refractive index, having uniform or equal refractive index to polarized light. So I band is from isotropic, it means having uniform or equal refractive index to polarized light. Right? So A band and I band, these alternate. You can see that A band or dark band contains myosin filaments and portion of actin overlapping the myosin. Portion of actin overlapping the myosin. I band or light band contains only the actin filaments. Actin filament. Now in the center of the A band, there is a lighter area. There is a lighter area in the center of the A band, which is called H zone. H zone. See, a lighter area in the center of the A band. H is from Hansen, German word which means light. It means light. So in the center of the A band there is a lighter area called H zone. H is from Hansen, a German word which means light. Right? Now in the center of the H zone there is also M9. This is the M9 in the center of the H zone. M9 in the center of the H zone. In the center of the I band, which is isotropic to polarized light. So in the center of the I band, a light band, there is ZDisc. ZDisc, you see. This is ZDisc in the center of the I band, a light band. ZDisc gives attachment to actin filaments on two sides. ZDisc gives attachment to actin filament on two sides. You see, this is the ZDisc giving attachment to actin filaments on two sides. On two sides, right? So A band is also called dark band. I band is also called light band, and these alternate. Dark and light band alternate, and these give transverse striations or cross striations to the skeletal muscle. You see, this is the thick filament or myosin filament, and this is the actin or thin filament. Right? This is the ZDisc. This is the ZDisc. You see, small projections come out from the sides of the myosin filament. You see, small projections is come out from the sides of the myosin filament. You see, this is the myosin filament, and small projections come out from the sides, both sides of the myosin filament. These projections are called CrossBridges. These are called CrossBridges. You see, these are small projections from the sides of the myosin filament, and these are called CrossBridges. CrossBridges. So this is the arrangement of these filaments in the myofibril. Right? The actin filaments partially overlap the myosin filaments, and then you see these form alternate A band and I band. Right? ZDisc. Z is from a word Zwischenschäbig. Z is from the word Zwischenschäbig. Zwischenschäbig. It means between or disc, between or disc. So Z is from the word Zwischenschäbig. It means between or disc. ZDisc. In all the myofibrils or in all the muscle fibers is continuous. Is continuous. So ZDisc of all the myofibrils, all the muscle fibers are at the same level, and these are continuous. And this gives the transverse or cross striations to the skeletal muscle. Because ZDiscs of all the myofibrils, all the muscle fibers are at the same level, are continuous, and these gives the transverse or cross striations to the skeletal muscle. The portion of the myofibril or the muscle fiber between two successive Z-discs, right? The portion of the myofibril or the muscle fiber between two successive Z-discs, one successive other. This portion is called sarcomere. So sarcomere is the portion of the myofibril or the muscle fiber between two successive Z-discs. One Z-disc, the other one. And the portion between these two Z-discs is called sarcomere. Sarcomere. Sarcomere is the functional unit of the skeletal muscle. Sarcomere is the functional unit of the skeletal muscle. And sarcomere, it undergoes shortening during muscle contraction. It undergoes shortening during muscle contraction. It undergoes shortening during muscle contraction. You see, sarcomere consists of a band in the center, and then portion of I band at the end. Portion of I band at the end, right? This is sarcomere in the center, A band, and then portion of I band at the end, right? The length of sarcomere at rest is between 2 to 2.5 micrometer. The length of sarcomere at rest is between 2 to 2.5 micrometer. After muscle contraction, it may decrease to 1.5 micrometer. So after muscle contraction, it can decrease to 1.5 micrometer. So at rest, length of sarcomere is 2 to 2.5 micrometer, and then after contraction, it decreases to 1.5 micrometer. In the muscle, there are certain proteins. In the muscle, there is myoglobin, which is heme-containing protein. It has high affinity for oxygen. It has what? High affinity for oxygen. It can bind oxygen at a low PO2. It can bind oxygen at a low partial pressure of oxygen. So it can bind oxygen at a low PO2. It acts as reservoir of oxygen. It acts as reservoir of oxygen. This was about the myoglobin in the muscle. In the muscle, there are also contractile proteins. In the muscle, there are contractile proteins, and these include myosin, actin, tropomyosin, actinin, actinin, and titin. So these are the contractile proteins in the muscle: myosin, actin, tropomyosin, troponin, actinin, and titin. I again repeat: myosin, actin, tropomyosin, troponins, actinin, and titin. These are the proteins present in the skeletal muscle. First, about the myosin. Now keep in mind that in each myofibril, there are 1500 myosin filament. In each myofibril, there are 1500 myosin filament and 3000 actin filaments. So in each myofibril, there are 1500 myosin filaments and 3000 actin filaments. In the myosin filaments, the protein myosin is present. So in the myosin filament, the protein myosin is present. And there are 200 molecules of myosin. There are 200 molecules of myosin in each myosin filament. So in each myosin filament, there are 200 molecules of myosin. So in each myosin molecule, there are 200 molecules of myosin. Each myosin molecule is composed of two heavy chains and four light chains. So each myosin molecule is composed of two heavy chains and four light chains. Molecular weight of myosin molecule is 480,000. So molecular weight of each myosin molecule is 480,000. In each myosin molecule, there are two heavy chains, each having a molecular weight of 200,000. So in each myosin molecule, there are two heavy chains, each having a molecular weight of 200,000, and four light chains, each having a molecular weight of 20,000. So four light chains, each having a molecular weight of 20,000, and this makes the molecular weight of each myosin molecule, that is 480,000. So two heavy chains, each having a molecular weight of 200,000, and four light chains, each having a molecular weight of 20,000. The two heavy chains are wrapped together to form a double helix. Each heavy chain, two heavy chains, are wrapped together to form a double helix, double spring. So two heavy chains are wrapped together to form a double helix. At one end, the heavy chains are folded. At one end, you see, the heavy chains are folded to form the head portion. So at one end, the two heavy chains are folded to form the head portion. And in the head portion, you see, in the head portion, there are 4 light chains. So 4 light chains are present also in the head portion. So two heavy chains are wrapped together to form a double helix, double spring. At one end, the heavy chains are folded to form the head portion. In the head portion also 4 light chains are present. There are 3 parts of the myosin molecule. 3 parts of the myosin molecule. Tail or body, arm and head. So tail or body, arm and head. These are the 3 parts of the myosin molecule: tail or body, then arm and then head. The arm and head portion is called CrossBridge. CrossBridge. Right? You see the myosin molecule having small projections from its sides, and these projections are called CrossBridges. What is a CrossBridge? CrossBridge is the arm and head portion of the myosin molecule which projects out from the side of the myosin filament. So the body or tail portion of the myosin molecule are present in the central portion of the myosin molecule, while the cross bridges, that is the arms and head, these project out laterally. These are the cross bridges. This is the central portion of the myosin molecule having the tail or body portion of myosin molecule. So you may be very clear about the cross bridges. Cross bridges protrude out from the sides of the myosin molecule, and these consist of the arm and head portion. This body or the tail portion of the myosin molecule are present in the central part of the myosin filament. In the myosin molecule, there are 2 points at which the molecule is highly flexible. So there are 2 points at which the myosin molecule is highly flexible. You can also see the 2 heavy chains that are wrapped together to form a double helix, right? The tail portion, the arm portion, and the head portion, right? So I told you that there are 2 points at which the myosin molecule is highly flexible, and these are called hinges. These points are called hinges. Kabze, hinge, kabza, the hinges of the door. So there are 2 points at which the myosin molecules are highly flexible, and these are called hinges. So one hinge is between the tail and arm, and the second hinge is between the head and arm. So one hinge is between the tail and arm, and the other hinge is between the arm and tail. So at these 2 hinges, the molecule is highly flexible. Right? In the myosin head, there are two important sites. In the myosin head, there are two important sites. This is the myosin head. It has got two important sites. One is actin binding site, which binds with the active site on the actin filament, which binds or which interacts with the active site on the actin filament. And the second site is catalytic site. Second site is catalytic site having ATPase activity. So catalytic site having ATPase activity, which can hydrolyze ATP to ADP to liberate energy. So two sites in the myosin head: actin binding site, which can bind, which can interact with the active sites on the actin filament. And the second site is the catalytic site having ATPase activity, which can hydrolyze ATP to ADP to liberate energy. So this was about the myosin molecule. I told you in each myosin filament there are 200 molecules of myosin. So this was about the myosin protein present in the myosin filament. Now we come to the other proteins, actin. So actin protein forms the basic skeleton of the actin filament. It forms the